Hello, my name is Leticia Wasa. My leg number is 23 19811. I'm going to present about the I'm going to present about the assignment I have 5 questions from 36 up to 40. I'm gonna start with the question number 36, which is about the difference between sandwich ELISA and indirect ELISA. Sandwich ELISA. Well, here in the sandwich ELISA, the first thing is for sandwich ELISA, we detect the antigens. Here in the principle, they are saying that the well is coated with the capture antibody that binds to the antigen. Here, the antibody binds to antigen. After the sample is added, an antigen present is bound by the this capture antibody. A second antibody, which is linked to an enzyme, then is used to bind to the antigen, forming a sandwich of the antibody and antigen antibody complex. Here in the sandwich ELISA, we use two antibodies which bind to the antigen and they form a complex. Here in this, they are telling us the advantage of this sandwich ELISA, which are the sandwich ELISA is a highly sensitive as the antigen is captured between the two antibodies, increasing the specificity. And they use where we use a sandwich ELISA, we use a sandwich ELISA to detect a large molecules such cytokines, hormones, and viral proteins. And we move on to the indirect ELISA and the principle of this. As we know, indirect ELISA is used to detect the antibodies in the sample, and in the principle they are telling us the well is coated with an antigen and the sample containing antibodies is added. Here the sample is coated with antigen, and the sample containing antibodies is added after washing away and burnt antibodies, secondary antibodies, which is linked with enzyme where the substrate is going to bind to make sure that any there is their antibody is added to bind the primary antibody. The secondary antibody bind to the primary antibodies, which is detect the color change. And the advantage of this indirect ELISA, we have simple and cheap. This indirect ELISA is cheaper compared to the sandwich ELISA and is used in serological tests to detect the presence of antibodies against the pathogens. Here they are telling us where we commonly use indirect ELISA. They are telling us that we use indirect ELISA in the diagnosis of infections like HIV or hepatitis, as well as in research. We also use indirect ELISA in research. We move on the second question, which is 7, which are asking us the application of ELISA in clinical practices. ELISA is used in different ways. Like the first thing where we use the light ELISA is used in infectious disease diagnosis. ELISA is used to detect specific antibodies or antigen related to the diseases such as HIV, hepatitis, malaria, and COVID-19, also tuberculosis. Have hormone and protein measurements. ELISA is used to detect to measure hormone level, example like insulin and thyroid hormone, cytokines, and other proteins in conditions like diabetes, cancer, and autoimmune disease. And the third one, we have safety testing, where the ELISA is used to detect any contaminants in food such as proteins and pesticides. And here we use the ELISA in cancer monitoring, detect tumor markers such as CAI to monitor cancer progression and response to the treatment. Here and number 5, we have allergy testing and identify specific allergens causing allergic reaction by detecting IgE antibodies against a certain environment of the allergen. And we move to the third question, which is 38, which is which is about the main types of vaccines and their characteristics. We have different types of vaccines. The first one is live attenuated, which is characterized by weakened form of pathogens and stronger immune response, lifelong immunity. Here, in the example of this dear live attenuated vaccine, we have yellow fever and retrovirus or MMR. This live attenuated vaccine is a weakened form of pathogen and they have a stronger immunity. And we move on the inactive or killed, which is dead pathogen, weak immune response, and booster the doses needed. And the example of this inactivated killed, we have polio, hepatitis A, and rabies. And we move on the next one, which is recombinant and subunit or conjugate vaccine, where pathogen fragments and no live components. We have a also safe for immunocompromised people. And this example, we have hepatitis B and pneumococcal. And the fourth one, we have toxoid, which inactivate the toxins and neutralizing antibody produced. And we have the example, which is the tetanus. And also we have messenger RNA, where messenger RNA instruct the cell to produce antigen and no live viruses. And the example we have COVID-19 as the example of messenger RNA viruses, now messenger RNA vaccines. And then we have DNA, which is characterized by DNA plasmid encode antigen and stimulate immune response, where we have COVID-19 as an example. We have virus like practice, where mimic virus, no infection, stronger immune response, where we have example as HPV and hepatitis B. And we move on the 39 question, which is asking about the heredity immunity contribute to the prevention of disease. And hereditary immunity occurs when the significant proportion of population becomes immune to an infectious disease, either through vaccination or previous infection, thus reducing the spread of disease. The key points include prevention, spread, protection for vulnerable individual, and threshold of immunity. Here in prevent spread, where enough individuals are immune, it creates barrier that limit the ability of pathogen to spread. And the second one, protection of vulnerable individuals, people who cannot be vaccinated, like infants or immunocompromised individuals, are protected because their diseases are less likely to spread. And here in tertiary, they are telling each disease has tertiary of immunity needed to prevent transmission. For example, measures requires about 95% of the population to be immune to stop our outbreaks. And we move on the last question, which is difference between active and passive immunity. Where active immunization, the immune system is permitted to produce its own antibodies against the antigen after exposure or to a vaccine or infection. Here, the body stimulates its own antibody against any exposure or any infection. And this active immunization take a long-lasting immunity and often requiring a few weeks to develop. And examples of vaccines, we have influenza vaccine. And passive immunization, you have principal performance antibodies are directly transferred to any individual, providing immediate immunity, and duration it provides short time of protection. Example, we have monoclonal antibodies and the mother's milk. I think this is the end of my presentation time.